And our next speaker is also from far away. From Fateme Babaha is from David Lillicrop's lab at Queen's University.
Good afternoon, everyone. First, I would like to thank organizers for providing me this opportunity to present my research. Once again, my name is Fateme Babaha from Queen's University. Our lab focuses on gene therapy in hemophilia A, and today I'm going to talk about evaluation of a novel native enhancer element from the factor VIII locus to improve AAV-mediated factor VIII transgene expression. So hemophilia A is a X-linked recessive bleeding disorder caused by pathogenic variations in factor VIII gene, which encodes factor VIII coagulation protein. There are several variations involved, and all of them will result in the reduced factor VIII coagulation activity in the plasma. As seen in the figure on the bottom left-hand side of the slide, it shows the role of factor VIII in the intrinsic tenase complex of the coagulation cascade and how it facilitates the conversion of factor X to factor Xa, which is integral for proper fibrin clot formation. Patients with hemophilia A experience recurrent and spontaneous bleeds into muscles and joints, and currently the treatment available is actually prophylactic, or in response to a bleed. So the approach that actually offers cure for patients is gene therapy, which provides them with endogenous expression of factor VIII. In hemophilia A gene therapy, recombinant AAV, or adeno-associated virus, is used to introduce the working copy of factor VIII. To do so, rep and cap gene of the wild-type AAV is removed and replaced with factor VIII cDNA, along with a liver-specific liver-specific promoter, as well as other regulatory elements. This transgene expression cassette is then packaged into an AAV capsid, which can take no more than 5 kb of the DNA. So this really imposes a challenge in designing expression cassettes, as the factor VIII cDNA, once it's B domain deleted, is 4.5 kb itself. So there is very little room for the regulatory elements that can be incorporated within the expression cassette. Currently, clinical trials of hemophilia A show a reduction of factor VIII expression and subsequently factor VIII activity levels following a peak at around 6 months, which remains at low levels for the remainder of the study. One of the possible mechanisms could be reduced mRNA production or insufficient transgenic protein trafficking and secretion. Two strategies that can be done to overcome this is to use hyperactive forms of factor VIII, which has improved activity and secretion, or use minimal regulatory elements with high transcriptional activity. On the top of the slide, we can see factor VIII PAJA, which is a hyperactive form of factor VIII, which is actually a large duplication within factor VIII gene, which is shown to increase factor VIII mRNA transcription and factor VIII translation. On the bottom, you can see in silico analysis of about 1 kb of this region, which has marks of active transcription, like DNA signals and peaks of transcription factor binding sites. For the sake of our study, and considering that in the future we are going to use this regulatory element in an AAV vector, we selected a smaller portion of this sequence, about 400 base pair of it, and we aim to investigate its enhancer potential by doing functional assays. On the top, we can see that this enhancer element was cloned into a reporter plasmid, and then it was transfected into various cell lines, and as seen in the figure, the enhancer plasmid could increase the luciferase activity within all cell types. On the bottom, we can see animal studies of which the same enhancer plasmid was infused into mice, and 48 hours later the mice were monitored, and as seen in the figure, mice which received the plasmid containing the enhancer actually did express luciferase. So the main finding of this study is that plasmid construct with regulatory sequence derived luciferase expression in cells and study mice in both orientation, confirming its role as a ubiquitous transcriptional enhancer. Currently, we are now working on reducing this enhancer element to less than 200 base pair, and we are actually doing the same set of experiments to further confirm that this smaller sequence also plays a role as an enhancer. And once we are done with that, we will proceed with the development of AAV vector, and we will do the gene therapy in mice. And with that, I would like to end my talk, and I'm happy to take any questions.
Thanks for that talk. My question is about the adeno vector. With the use of the adeno vector for vaccines for COVID, it exposed the risk of vaccine-induced immune thrombotic thrombocytopenia, which led to pretty much the discontinuation of that platform right now by Johnson & Johnson AstraZeneca. How does this impact gene therapy using these vectors? Will there be resistance because of the fear that VIT will happen? And on top of that, we've recently shown that adeno infection can lead to a VIT-like syndrome too. Do you think that's going to impact the vehicle?
Thank you for the question. Well, actually, the vector that we are using is adeno-associated virus and not the adenovirus itself. It's less pathogenic in humans, so I don't think that we will see that much of an immune response. After infusion, we will see immune responses following vector administration if the vector dose is very high, but the AAVs itself is way less pathogenic than the adenoviral vectors. Yeah.
Thank you for your talk. One of the things I think we've seen in hemophilia is the difficulty with anti-AAV antibodies affecting transgene expression long term. Is this method something that can be used with other vectors, or would it mitigate that risk in some way? I don't think so, but I figured I'd ask.
Well, that's a very good question. Actually, naturally, we are exposed to AAVs in early life, so there, like about 40% of the population has the inhibitors or antibodies against the AAV capsid. So in the gene therapy trials, one of the exclusion criteria is presence of the antibodies against the capsid. But for now, the types of viral vector that is used for gene therapy in hemophilia A is still AAVs.